Hello, good morning, everyone. So we're just gonna wait a few more minutes till more people join, and then we can get started with compensation today. I see a raised hand. I'm not sure if you can unmute yourself, but in any case, you can write your question on the chat box if you want. There is somewhere a chat box or Q&A box. So feel free to use that. I'll try to keep it to watch it regularly for any question or anything you have during the webinar. Yeah. Okay. Just to say, this webinar will be recorded, and of course, we always have it available afterwards in our website. So I can show you once quickly where you can find those webinars. So if you go to flowjo.com, okay, exactly the same way that you found this webinar, right? You can go here, webinars, under the Learn tab. And then right down here, we have the recorded webinar. So if you click on here, in a few days, maybe tomorrow, maybe on Friday, maybe next week on Monday, this webinar will be also right here. Yeah, so don't worry. We do it as fast as possible, and then you can watch it on demand anytime. Okay. So let's get started slowly. So I see more. Thank you very much for joining. So today we're gonna talk about compensation in FlowJo software version 10. My name is Ioannis Panetas. I'm a senior scientific advisor at FlowJo Informatics, which is part of BV Biosciences. My email address is just here. Yeah, there is a lot in compensation we're gonna see today. I hope I don't overwhelm you. But if you have questions also after the webinar, or if you try on your own data to compensate and mix or whatever else we're going to be seeing here, feel free to email me with any question and I'm happy to take it. And the same thing applies for the chat box. If you have any question throughout the webinar, feel free. I'll try to frequently, sporadically check any question and address them here live. Okay. So as I said, we're going to dive into compensation. So this is a topic that becomes, of course, more and more relevant in flow cytometry, given that flow cytometric experiments tend to increase nowadays in complexity, as well as having new instruments with more detectors, more dyes, which are more specific for a specific um, uh, region of light, so to say. And so all those factors allow us to also bring our research much more in a much more higher complexity with multicolor experiments. And although you might think that the process of compensation is only something you can do on the cytometer, the truth is that even after acquisition, within the FlowJo software, we can perform compensation right from scratch. Yeah. I was also, when I was using the cytometer in my lab, I was always doing the compensation with the instrument. That's not wrong. Of course, it's not wrong. It's just there is many things we can do post acquisition, and this is what I want to address here, and not just visualize this matrix from the instrument, but also modify and create a new matrix, as well as having different metrics of goodness of the compensation matrix, which we're going to be talking later. Okay. So what's on the menu today? Let me use this pointer here. Always getting lost with this zoom bar. Okay. So we're gonna start with the very basics of what is compensation, and specifically when and why would you need it for your experiment. Then I will show you some specific tools and algorithms that we have in FlowJo for calculating the compensation matrix. So I will show you the full workflow and the different choices you can choose to make it easier for you to compensate. Yeah? Finally, I will introduce in the second chapter to some metrics. So we can use different metrics in order to evaluate our compensation matrix. Also, you might have heard previously of the spillover spread matrix, the total spread matrix, and other statistics that we can calculate in order to see how good or bad is our panel, or to simply measure our instrument performance in case of the spillover spread matrix. So I'll show you those in detail and when to use, which one to use, and also I'll show you a new metric that we have, which is available in the BD Research Cloud platform for free, and it's the hotspot matrix, yeah, which address more specifically the unmixing spreading error in spectral map, yeah, and how easily you can predict which spots of your panel need to be improved. And finally, I'll just show you some common troubleshooting tips. So you might have seen shapes like that after compensation. I'll show you what is the most common and most frequent errors that we do in terms of single thing controls that we get such a result, and how we can correct that within FlowJo. Okay. I'm sorry, I just lost again my Zoom bar here, which is somewhere hidden. Ah, there it is. Okay. Very good. Okay, here we are. Now, let's get started with some introduction to compensation and theory, right from the beginning. So, to understand what is this complex term of compensation deeply, we need to understand first of all what it's made from. Yeah, why? What is causing this need that we have to compensate before performing an experiment? So flow cytometry was, of course, invented many decades ago and embraced the use of fluorescent compounds. Yeah, so fluorescent compounds have this property of absorbing light energy and causing electron excitation. Yeah, so if we have an electron of this fluorescent dye, here we have 50, for example, it would absorb light. Yeah, so in that case, it would absorb light at the 488. It would absorb this light, and then it would bring the electrons to the excited state. Yeah, from this now excited state, we have some energy loss, which is not the cause of the emission yet. But after this, we have the actual emission and energy loss to bring back here the fluorescence of Fiji, yeah, which excites at around 530 here. What we see here. This diagram is the emission of Fiji upon excitation with a blue laser. Yeah, so what is actually happening here is that we have those green photons, and after this observation, of course, scientists wanted to conjugate those fluorochromes or fluorescent compounds with monoclonal antibodies. Yeah? And that would be an ideal way of measuring some specific characteristic of a cell. Yeah? So, for example, if we would conjugate Fiji with CD3 antibody, we would be able later on to, after we excite the cells with the 488 laser, the blue laser here, we would be able to see, to detect this emission on cells that bound this monoclonal antibody and therefore have CD3 on their surface. So, and of course now that was a great way to bridge the emitted light with the biology and the presence of a certain protein on a cell. Yeah? So what we have to do is basically when we perform flow cytometric experiments, we need to isolate the cell population of interest. And I'll just show you a simplistic example here. Yeah, so we have, again, what we said, CD3 monoclonal antibody conjugated with FITC. And then we would try, in order to detect CD3 positive cells, we would try to excite with a blue laser the cell homogenate, and then we would have a specific detector. So here, this rectangle here is, let's say, a detector of the flow cytometer, which detects exactly this FITC fluorescence. Yeah? So now, from the emission, we can actually go to identification of these cells. Yeah, so from this detector, now we would have a signal which shows some cells that bound the CD3 that have this fluorescence of FITC. Yeah, so that was all easy. Yeah, the times that we had one color or two colors are very far away in this in their emissions, that was a very easy job. Yeah, but let's see another example now. Yeah? So let's say we wanted to go more deeply to different cell types, yeah? And we have not only CD3, but we also want to identify B cells with CD19 and PE here. Yeah? So we would add both antibodies. So CD3 would bind T cells, CD19 would bind B cells, so different subsets. But what we can observe now here is that when we excite with the same laser, the blue laser, we would have an overlapping emission. Yeah, so you see here, when we excite FITC with the 488 laser, we see that some of this emission of this antibody here overlaps and spills into the secondary detector, which is the one that detects PE, yeah, and CD19. Ideally, what we would want in this experiment would be to identify our B cells and our T cells like that. So we have one population here and one population here, so different colors basically. But what would happen without compensation is something like that. Yeah, so we would see that our CD3 positive cells appear in this, this banana shape. Maybe you see it sometimes, which seems, which is the first sign that your data are uncompensated. Yeah, so some of the FITC here spills into the PE channel, and therefore they appear double positive. But that's not the biology behind it, right? So it's this spectral emission spilling into PE that we need to find a way to correct. Yeah? And we're going to jump now next into how we can correct this spectral overlap between different detectors. Okay. So this phenomenon, as I said, you can hear it with many different ways. Spectral spillover, spilling, signal spilling into secondary channels, etc. So all in all, spillover refers to this overlapping emission spectrum I mentioned before, from one fluorochrome to different detectors down the primary. Yeah, if it was only the primary, we would never, need compensation. The sources, unfortunately, we cannot always exclude that we have detectors close to each other. We need to take all light and partition it, but there's not so much we can do to split even more the light. Right? So we cannot avoid having detectors close to each other. Some dyes are also cross-excited by different lasers. Yeah? And other sources would be residual donor fluorescence, like for example, here PE and PE size 7. Of course, you would still have some PE in the PE size 7. That's why you would need more compensation by using a panel with those two dyes. So let's look at now at the more specific example of how this spectral spillover can affect data interpretation. Because again, we're at FlowJo here, so we want to see what we would expect to see when we have compensation and when we don't have compensation. Yeah. So another experiment here. We have, let's consider a panel which consists of two fluorescence antibodies. We have CD4 in PE Texas red and perforin in PE. I'm not saying that this is a good panel, of course, but if we had two colors, we would avoid using those two, right? But this was actually an example from a multicolor experiment that we didn't have so many choices, right? So if we would do that, we would have some CD4 stained cells and some perforin stained cells. Yeah. So what would happen is what we explained earlier. We would have PE spilling into PE Texas red, but also the opposite. PE Texas red, we see it spills also into PE. So now we have two-way spillover from primary channels to secondary channels. How would we see that in FlowJo? We would see something like that, right? We would see two double positive populations right here. So whenever any cell is positive for any of those two parameters, it would be also positive for the other. Okay. So of course we wouldn't want to gate like that, right? We wouldn't want. I don't have the confidence myself here to place a gate and say those are CD4s and those are perforin positive. Yeah. So first of all, we would need to compensate in order to safely make conclusions and draw gates to identify those populations. Yeah. And now another example, we could actually do something more clever, in that case for panel design, where we would use, instead of P and P Texas red, we could use CD4 Alexa 488 and perforin Alexa Fluor 647. And in that case, if you look at the spectral spillover, you'd see that those two have a very low spectral spillover, almost none. Yeah. So in that case, actually, we can interpret that data much better. Yeah? So you can see quite easy without any compensation. We have a CD4 and a perforin positive population. Yeah. But again, there is little we can do if we use a multicolor panel. We would need to make hard choices of, either fluorochromes that overlap too much to each other, or you have to at some point, right? You cannot have only mass cytometry. You cannot have all these. Yeah? In fact, it turns out that we can correct. Yeah, so this spectral spillover from PE, Texas red, and PE, if you apply compensation, okay, you can bring that to how it would be if you have a panel where there is no spectral spillover. Okay, and this is what exactly is going to be our topic of today, which is compensation. Okay. So all in all, with compensation, you can imagine if you're a bit more artistic, we go from a complete spectral view of all colors, okay, which can, for example, represent a final painting, which our cytometer recognize the full painting, so to say. And what we need to do is to instruct the cytometer how to go back from the full painting back to the single colors. Yeah, we need to break it down to the contribution of each antibody in each color in the final fully stained sample. Okay. Okay, so compensation is this process, going into a bit more scientific terms, rather artistic. Compensation is the process of determining how much of the signal is emitted into the secondary detector by one fluorescent lamp. Yeah? It has to do with the fluorochrome selection and the cytometer. Yeah, how many detectors, lasers, and so forth. Yeah, so it doesn't depend on any cell type or probes and so forth. It's only a factor that contributing there is the fluorochrome selection and cytometer. So what we need to do We need it in order to determine the true amount of light that is coming from a single fluorescent dye into a specific detector. Yeah? And then, as I said before, we can distinguish if our cells are truly single or double positive for a marker. Yeah, so here below you can see an example from the BT spectrum viewer, a panel with FIT-CPE, PER-CP Sci5 and PE Sci5. Yeah. So what you would see before compensation for those two here, okay, you would see something like that. And upon applying compensation using single stain controls, this blue population would have the same median. Okay, we need to bring negative and positive to have the same median for PE size 7. Yeah, so this is what we would observe. You see that spreading here of the population. This is something we're going to talk later about, the spillover spread. So what I can say now to you is that it's not compensation that makes that. Yeah, the compensation only reveals that spreading. Yeah. So don't blame your compensation for that. It's the selection of fluorochromes or the cytometer configuration that would make this spread appear. Yeah, it's simply the error of detectors from fluorochromes, detecting fluorochromes with high spectral spillover. Okay, we're going to talk about this later, and especially much more during the metrics of the compensation matrix. Okay, just checking here also the chat box. Okay, let me know if you have any. Okay. Single stain controls. Yeah, you might have heard that for compensation. They're, of course, the alpha and omega of for compensation. Because we need a way to instruct our cytometer, whatever cytometer we have, the cytometer needs to know the individual proportions of its fluorochrome before running the multi-stain sum. Yeah, so if we have here, for example, I see 6 single color controls, single stain controls. If we have a 6 panel, we need first to add a tube from each one of the single stain controls so that we instruct and train cytometer how to distinguish each one of those fluorochromes. Yeah, and in addition, you will also need an unstained sample. You'll see many applications here today of how we can use the unstained sample either to remove autofluorescence or to establish what is the background fluorescence of your samples. The unstained control will be very, very crucial for that, yeah, because we need a negative, but the cytometer also will account for calculating this matrix. Okay. Now, so I have a very simple 3-color experiment here, yeah, that I want to illustrate to you and talk about how we actually calculate this spillover coefficient and compensation. Yeah? So this compensation matrix. So here I have 3 different fluorochromes attached to a cell, right? And we have 3 different detectors. So I have my, you might see here this darker detector. This is what we assign in the instrument. Yeah, we assign, we have this fluorochrome 1, and we say, okay, this fluorochrome has the highest emission in here, so I assign it to have the, the upper hand in measuring that fluorochrome. And the other 2 are secondary detectors now. And the same happens in the fluorochrome number 2. We have the second detector here, and the other 2 are secondary and or non-primary detectors, and the third one like this. Yeah. So here you can see an example of the fluorochrome 2. Okay, it spills over into detector 1. Okay, you see this emission here, and to detector 3, apart from the detector 2, which is the primary. Okay. Now, in order to calculate the compensation, So if we rotate this graph and we have now each fluorochrome as a column and each row as a detector, okay, we're actually, if we reverse this matrix, we can do some very complex calculations here. Okay, so if you translate this into a matrix here below, you will assign, for example, those spillover values here on the matrix. So you see here, for example, detector 1, we assign the number 1 here for that detector. We have a spillover value of 0.15 here on for the fluorochrome 2 on that detector, and the same for fluorochrome 3, we have 0.01. Okay, so what we actually want here is to solve this linear equation. And I cannot tell you how difficult that is to do it by hand. Yeah, I would really avoid doing this by hand. You know, my father is mathematician, and I asked him, okay, how could I solve something like that? I couldn't. I thought it would be easier, but it's very complex to find, you know, the contribution of bits. This compensation matrix is essentially the reverse matrix of this spillover, okay, that we need to multiply by in order to find each single fluorochrome fluorescence in a detector. Yeah, so this takes a very hardcore algebra calculation in order to have. Yeah, so that's why we would prefer to have algorithms to find this matrix. And then what will happen is when we calculate this matrix, this matrix will basically remove all the contribution of the fluorochromes to the secondary detectors. So, and the same way, okay, so then if we apply this matrix to our fully stained samples, we will know that we don't have any contribution of spillover into the different non-primary detectors. Okay, and something very, very similar that you might have heard, okay, is the spectral data. Yeah, this basic example is the same exactly experiment, having 3 different fluorochromes, but in the spectral experiment, we have much more detectors. Yeah, so we still calculate this spillover matrix for all detectors. The difference is in approaches is that you have a much wider space with different filter sets, and all those filter sets are designed much more sensitive to only a fraction of light. Yeah? But the calculations are similar, a bit, of course, more complex. But instead of having 1-to-1 ratio of one detector to one fluorochrome, we have multiple detectors where we compensate the fluorochrome across all detectors. So we have a rectangular matrix right here. Okay. So let's see now the different compensation methods we have. Yeah. So starting with the traditional method, it's the one that you would do also in your most of the cytometers in the instrument. So how can we do that in FlowJo if we haven't, for example, compensated the instrument, or if we want to do it from scratch? Yeah. So what we will do first is we will add our samples, our single stain controls, including the unstained sample, into the compensation. You can click on the algorithm here on the top right after you click on the compensation wizard and select the traditional spillover algorithm. So in what will FlowJo try to do is to match your fluorochromes to the each one of the single stain control to the parameter that you have in your experiment. It will create some gates where it finds the highest fluorescence, and then what you need to do is to assign to designate positive and negative gates. Yeah, so this happens right here on the right. Yeah. So you need to select a negative gate. So this could be your unstained controls or in-tube negatives, whatever you have. And the same way you have to find your positive cells. Yeah, so what you would have ideally is you would have two different peaks here, a negative and a positive. Okay, the blue one is the negative, green one is the positive. And here you need to place a gate, yeah, for each one of your single stain controls. Okay. Again, so when the computation begins, FlowJo will create this gate called cleanup gate or a size gate, but you can also have your own gate. We don't want all of the cells in a sample in order to calculate the negatives and the positives. We can filter to the population that we expect the expression of this specific antigen. Yeah? So we don't need here to have all PBMCs and monocytes if it's a CD3 marker. Okay. But this FlowJo will do by itself. I'll show it to you later, a bit later in the demo. And then FlowJo will create also some exemplar gates for negative and positives. Yeah, so you can correct them if they're, FlowJo tries its best to do it so that you don't have to manually adjust it. But sometimes you might, especially if your antigen is not expressed that much, you might need to resize or readjust those gates. Yeah. Then the spillover coefficients are calculated with the process we explained before, and they're inverted in order to bring this population here to have the same MFI with the negatives. Yeah. So this is what will happen. We will have a single positive population, and again, how I mentioned earlier that we do have this. Sometimes, but depending on how close those emissions of those two fluorochromes are, we might meet this spreading error. Okay. Okay, so let's get into FlowJo. Before I move to the next algorithm for compensation, I would like to show you first the basics. Yeah. So let me just quit here the presentation just for a moment and go into FlowJo. Okay, so I'll open here FlowJo. And for here, I'll use some conventional data. Yeah, so this will be my traditional compensated data. Okay, and I'll just load those samples within FlowJo. Yeah. So what happens here is you will notice that I already have 12 samples that went directly into my compensation. Yeah. So if you wonder how and why that happened, how did FlowJo recognize automatically which samples to put in the compensation group, you can go always to the preferences. So in the preferences here, in your preferences here, you can go under compensation, okay, and you will see those inclusion rules. Yeah. So basically, FlowJo can recognize specific keywords to automatically transfer your single stain controller files within the compensation. In that case, the keyword used is the $FREL, which is basically the name of the file. So whenever FlowJo looks at the names of your file and it identifies beads or comp or unstained or single or whatever else you want to add in here, it will automatically put it in the compensation group. Yeah, so that's also a good tip. If you whenever you prepare the names of your samples in the cytometer, remember to add some detail on what exactly is this sample. If it's a single stain control or comp, you can use some of those keywords so that FlowJo then will find them directly and we don't need to drag drop, etc. Okay. So I see a question here from Ralph. The question is, if your panel consists of marker for T cells and monocytes, yeah, how would you draw your gate for autofluorescence? Because autofluorescence differs between these two populations. That's a very good question. And so I think in that case, you could, so if you have a mixed panel with T cells and monocytes, yeah, then it might be that the your compensation matrix in the end would be, slightly different depending on how much autofluorescence you have in there. You might need to have different matrices. Yeah. So ideally, what I would do is to have many different controls where I have the same fluorochromes in different markers. So, for example, if you have PE in your panel, you could have, for example, CD11B to have monocytes expressed in PE and CD3 in PE to have for T cells. Yeah, and then you could input. So ideally, the more controls you have, I would add those to when I have to make conclusions about the how many T cells or what is the expression. So if you want, you could also, something you could do to bypass all this number of controls, maybe you could use a more generous gate where you could include both your monocytes and your T cells in there. Okay, so that then you have, as the background fluorescence, you have both together. Yeah, so then your matrix, I think, would be then could be applied much better into into both populations. So then you won't need a specific matrix for each population. Yeah. So I would rather use both a cleanup gate, including both monocytes and T cells. For another question is for the reference of papers. I'll show you those to learn the math. Yes, so I'll show you later in our website. There are some references for the traditional compensation. I'll show you in the FlowJo documentation later on. Okay. All right. Now, let's do the following. We have our single thing controls into the compensation group right here. Okay. What we need to do next is to press this compensation button. And when I press that compensation wizard, it will start the calculations. Yeah, so it will try to identify the size gate that I said and positive and negatives within each tube. Okay, so let's see what's happening here. I'll press the button. Okay, you see there are some calculations, and you see that it created everywhere this size gate. Yeah, so I identified a cleanup gate, yeah, and specifically within that gate, the expression of PE or Alexa 647 here, etc., etc. Okay, so in the main compensation window, okay, what we need to do next here, let's expand that a little bit. Okay, FlowJo also tries to assign the single thing controls to a detector. Yeah, in many cases, this is correct. Yeah, so the more detail you add into the sample name, also, it's much easier to find. Yeah, so here you can see the ARD was found correctly. So you see this green color. We have correct association, and you can start checking if the correct single thing control was assigned to the correct parameter here. Here we have a blank channel, so I can remove that. I can press right click and remove this parameter. So I think this was just left open. It's not, it's just PBMC. There was no really really autofluorescent populations, but I'll show you later how to deal with that. Okay. And the next step is, after we have the correct assignment here, for the traditional algorithm that you see I have selected here, the spillover algorithm, I need to assign a negative and a positive population. So if you have an unstained control, you can use your unstained control, okay, as the your unstained control as the universal negative. Okay? If you drag and drop your unstained control here, it will assign everywhere. Yeah? So here I have unstained beads and unstained cells. Okay. Remember that we always want to have the same source of sample. Okay, if we have beads for the negative, we need beads for the positive, right? So that's why here I have two different plots. So you see the negatives here, which is unstained beads, and the blue peak is the negative beads, and the green population here, the green plot here, corresponds to the positive for APC size 7. Yeah, so this looks correct. Yeah, so I can go scroll down and see how FlowJo did. Okay, here it looks fine. Here we have cells, so we have cells and cells, which is good. Bits again, and the rest looks fine. Yeah. However, I didn't show you this yet, but the very first parameter, which is the live-dead die, okay, we have a problem. Yeah, it's not a big issue, but what we see is that the blue peak here overlaps with the green peak. The negatives overlap with the positives. Yeah? And that, in fact, is happening whenever you, well, basically here we have mainly alive cells. That's the problem. We need to change that gate in order for the dead cells to show up here in the plot. So what I can do is I can open here my positive for ARD single stain control, and I can actually expand this gate. So we're actually looking for positive cells here. And where can I find them? Well, if you want, you can press the color map here for AARD and see that you see where the positive cells are. Those are the dead cells. So that's what we need to do. We need to expand that gate. Okay. And if I expand that gate, you start seeing that peak showing up, right? So you see that peak, those are my dead cells here. Now, ideally, okay, once you see those positive popping up, you can move that gate. Yeah, so I can go right here and I can move that gate to the positive. So here are my positives. Okay. Here are the dead cells. And remember, when you do something like that, okay, you can, you should, as I'm gonna mention also later, you should also adjust your negative gate. Yeah. So here, okay, the difference is minimal. Okay, you see the background fluorescence of positive and negative is almost identical. But if I wanted to be really precise on my compensation, I would also have to change that gate. Yeah. This can be easy also if you copy it in the compensation group and add a new gate here, you can do anything and then assign it back here. Yeah. So ideally, you would want to have a similar gate so that you have the same background fluorescence between negatives and positives. Okay. Okay. So once we're ready here, and we have all the negative and positive assignments for each parameter and each single stain control, we can simply press this button, which is says here view matrix. Yeah, so I will name this traditional comp. Okay. And then I can press view matrix. There's a question, what does the green, yellow, sometimes red plots indicate at the left-hand side? Very good question. Well, you see here this green light for, let's take it, for example, those first, yeah, ARD and APC87. When the parameter name is included within your single stain control, which happens in these first two parameters, so we see exactly ARD, there's no mismatch whatsoever. Yeah? When it finds the exact same name, it will give you the green light that I found most likely the correct sample in here. Yeah? When you see this yellow, it means that there is a sort of mismatch in the name. So, and that's exactly here you'll see the notification. The file name of the selected sample does not appear to contain any references to this parameter. Are you sure it's the right sample? Yes, I am. It's just somebody got lazy in the cytometer, and instead of writing Alexa with AX, he wrote A488. Yeah, I would also write A488. I didn't know it's with X in there, but it's still okay. It's all good. It's all good. We just need to make sure it's the correct sample, but we're on the safe side here. Okay. I think it's the minimum for positive population. I think after, so what you can do to check that, whenever you see that the compensation is finalized here on the top right, it means that all percentages are fine. If you go here, if you want to quickly check that, I don't remember what is the border. Yeah, so below 2%, I had to do this to find out. 2% of the total. If it's less than 2%, then it's not going to be able to calculate. But yeah, that's for the traditional compensation route. For the AutoSpill, yeah, there's no minimum positives. Yeah, so for the AutoSpill algorithm, you bypass this issue. Okay. So let's now press View Matrix finally. Okay, and here we have our compensation matrix. Yeah. So here we see all the values. Below you can see in the end-by-end view, it's a parameter versus its other parameter. If you want, you can select even your fully stained samples and see if this compensation makes sense, or if you have any weird error appearing. And you can apply this compensation back to your samples. Yeah. So excuse me, just give me one moment. I'll be back in one. Okay, sorry about that. Yeah, so in order to transfer now this matrix back to my samples, I can simply go there. That's also very easy. You can go here and say, okay, apply this compensation, this traditional comp, into my whole group or my samples or any other group I have. Yeah. So then you would see that the matrix would change. Okay, and you'll see this red grid everywhere. Yeah. So this red grid, after I transfer my matrix from the compensation group, so here I pressed again. I think I was muted, sorry. After I press here apply to group. I can select which group I want, and I will apply this specific matrix into my full group here. Okay, and there is another open question here. Sorry, I just saw that. I just got the notification. How to choose the negative population to come from the stained single sample? That's a good question. So the question is, how can I have in-tube negatives? Yeah, let me show you this. A good question. Okay, so while I'm here, let's take this as an example, the live dead staining, and I want to have the same population right here for my negative. So I can simply go here up to the parameter of interest, go to negatives, and select the cell comp ARD, which is this one here. So when I press this one, you'll see that I have one plot. Yeah. So just so that you see the difference, I click. Now I have one plot with the in-tube negatives. Yeah. So I could also place a gate here for the negatives and use the same sample. Yeah. It's also correct. I'll show to you this a bit later with actually the AutoSpill algorithm, which where it's better to use in-tube negatives, okay, because it calculates a bit differently. You don't need those assignments of positive and negative. It calculates through linear regression. So it's better if we have the same sample and we compare the slope of positive and negatives within the same. So I'll show it to you in a minute. Okay. Any other questions for the traditional compensation? Okay, I'll check the Q&A as well. Okay. Okay, so let's move on with the next with the next subject. Okay, I'll go back to the presentation. Okay. Next topic. I talked a lot about it already, which is a good thing because I really like this algorithm. It's a different way of calculating the compensation. It was published by Carlos Roca a few years ago, and it's the AutoSpill algorithm. So it's a different approach in the way it calculates. So it uses linear regression, okay, to calculate the compensation coefficients. We don't need any positive or negative decades, so you'll see how much faster it is to use this algorithm rather gating, assigning positive negatives, then changing the gate. It's much more quick. Yeah, it's an iterative process, so that's very good because it optimizes. It doesn't have only one run and then it spits out the result. It performs iterative fitting of this line to fit all your data, and you have some benefits in subtracting autofluorescence. So you can use, for example, one of your unstained controls as a single stain control essentially, and the autofluorescence identified within your unstained sample can be compensated out. Yeah. The effect is everything is easier, and so is cytometry by using this algorithm. Yeah. So let me introduce you to AutoSpill. So what do we do here is the following. We have the same setup. We load our samples into the compensation group. We select then our single stain controls as we did before for the traditional compensation. And the only difference here is we will select the AutoSpill algorithm. Yeah. In addition to that, you'll see magically the positive and negative gates. The positive and negative gates will completely be gone. Okay, so we'll see something like that. And then in addition, you can select okay, the option here, remove universal negative. Yeah, so that way you won't have your Einstein sample and different tubes for the same parameter. You'll have the in positive and in tube negative and positive populations. Yeah? Then just to explain a bit the computation. So the spillover coefficients are calculated through this slope. Yeah, so we use a linear slope to find that line here that most of our data are on. So it finds, it adjusts this line iteratively to fit the whole data. And then it inverts that to create the same MFI as we said before for detector 2 and detector 1 and negative for positives and negatives, yeah, so that they have the MFI. So having no gates exactly means that we don't have a limitation in terms of controls where we have low expression. Yeah, so even if we have a control like that with very few cells as positive, the algorithm will still work nicely and calculate. And here is some figures from the paper just to show you the difference between traditional on the left and AutoSpill here. So in the traditional, we have by exponential display. You see here by exponential scale the data and how it is after compensation. So you see it here. The black dots are the post compensation. And in cases, so what they were saying for this figure is that basically AutoSpill works better when we have AutoSpill works better when we have controls with few cells. So here you see few cells and traditional. Okay, so we have these some outliers around. Also here, but with AutoSpill, you see how this line, when it's corrected, that it's much, much better compared to what you would have with traditional. Yeah? And that's exactly the way it calculates through that slope, which is reversed, and then we have this vertical line to correct for the compensation. Okay. If AutoSpill uses robust linear regression, which gives much more lower weight to the outliers, and that's also something very important that we don't have in the traditional compensation. So whatever is really outside of that line, it would be weighted less and excluded from the this slope. Yeah, and that's so that protects against any distortion in the data and also any autofluorescence. Okay. And as I said, this is an iterative process, so it happens step by step. So we have this line formed, and then it will try to recalculate. And when it reaches an acceptable point that this line doesn't change anymore, then it will stop and give you your results. Yeah. So let's go back to FlowJo and show you how this can be calculated. Okay. Quit here. Okay. So we were here. We can use the same example here. Okay. I can reopen my compensation matrix. Okay. And I'll show you how much easier it is to go to AutoSpill. So first of all, I'll change the name here because we're making a new matrix. AutoSpill. I can use a different color. Okay. So what I will do is I'll select here the AutoSpill button beside the algorithm. Click on it, and you saw the gates all left. Yeah. As I said, ideally, we don't want different samples for the same parameter, so I can click on Remove Universal Negative. So now I'm using the InTube. Okay, so what I need to be sure is that I have the correct sample, which has positive and negatives, and go through that list. And that's it. Now I can now I can select View Matrix here. It takes a bit longer, but it's relatively fast as well, because of this iterative process that it makes a lot of calculations. And once it finishes, it will give me directly the values. Okay. And that was it. Yeah. Now, in terms of values, if you wonder, because I know it might be something new for you, if you want to know what is the comparison between the traditional and the AutoSpread compensation, you can multi-select those two and see the differences. Yeah, and the nice thing is you can see even the differences here on the plots. So those blue are the uncompensated. The orange are AutoSpread, so it's a bit unfortunate color. I'll use maybe a blue one. Okay, so you can directly compare one-to-one those two matrices, but you see how similar it is. Yeah, the highest difference I see here is 0.3, which is really nothing for compensation. I don't see anything above 1, so it's faster. Yeah, I cannot say at this point that it's better matrix. Yeah. But you know, you saw how fast we can make a matrix and without really using universal negative and then controls, etc. Okay, and I can apply this matrix back to my sample. Okay. There's another question. Doesn't the light blue shaded region in the compensation matrix mean undercompensation? No. So what exactly? Let me go back so that I show you. The blue color is the uncompensated. Maybe you mistyped, you meant uncompensated. So yeah, the blue, this cyan color means uncompensated. Just here I said the blue color, maybe I could set a gray color or something. Yeah. So if I have red and gray, so you see one matrix in gray and one matrix in red color. But yeah, the uncompensated is with the cyan color that you can have in or okay. Okay, I see another question. I frequently use the SSM, but what is the CSM? The quotient similarity matrix. I will explain towards the end when we go to the metrics, if that's okay. And I'll show you. Yeah, in a few minutes, it's actually the next chapter here that we're gonna go into. Okay. Okay. So another question, if I understand correctly, for AutoSpill compensation, we only need the unstained universal negative control to perform the analysis. No, we simply mean we need a sample, okay, single stain control sample. Yeah, so we not completely unstained. We need, let's say you have a live dead staining as we had before. We don't want a completely unstained sample. We want a sample with a live dead stain. Okay, but and this we will input in there with, because not all cells would be dead, hopefully. You would have some negatives and some positives in there. Yeah. So having those two in the same sample, okay, AutoSpill can use that for the compensation. Yeah, it just there's need to be the single stain color panel. Okay. And the last question I will answer now: will AutoSpill AutoSpread work if there is only positive population in the control for CD45? No, it's not gonna work. So we need to have both. Yeah, at least even if it's few, I would really make sure that I have some, because if you have very few, if you have outliers, then those would be weighted less, and it might be that the calculations are, so you need to have some cells in there and not have only 2-3 cells. But even in the case of CD45, there's I mean, there's going to be some negative cells in there. Yeah, you can pre-gate your samples in order to find both and have a peak for both and use it later on. Okay, so I will continue here. Okay, with the next topic. Okay, which is autofluorescence. Yeah. So what happens with autofluorescence? And I know for some of you might be a very sensitive topic. For me, it was as well. I was also working with liver macrophages, which are extremely autofluorescent, like extremely in every channel. They were autofluorescent, and I realized back then that we need to keep, a channel empty with no antibodies. The channel with the highest autofluorescence, we keep it empty and just have it in every experiment. Yeah. So autofluorescence comes from molecular structure within the cell that naturally emit this light. Could be different, many different, that emit in different wavelengths. And of course, they interfere with the detection of the signal of targets. Yeah, so if you have a FITI antibody and your fluorescence is also in FITI, it's there it starts to become more difficult. Yeah. So the autofluorescence can be subtracted during compensation with unstained cells and an extra detector, the one that I told you earlier that we leave empty. Okay, there's two different ways, one for the traditional compensation, how to do it, and one for the AutoSpill. Okay, let me show you the two different ones. Okay, the first one is with the traditional. Okay, what is the problem with the traditional? Our problem is that if we have an empty channel, let's say here we have Alexa 647 as an empty channel that we want to input our unstained cells and remove autofluorescence. The problem in there is that the traditional algorithm needs positive and negative gates, right? So if I now add an unstained sample within my, if I assign an unstained sample on this parameter, I would only have a positive population. Yeah. So what we came up with is the true zero function, where basically this bypasses the fact that the algorithm needs some negatives and some positives. So what you can do is select here under the negatives on this unstained sample, you can select true 0, and this will calculate, it will have a 0 value for the negatives, and whatever is beyond 0 in this control, which is your positive gate, will be considered as autofluorescent. So the calculation will begin, and then the autofluorescence will be extracted from the rest of the channels or wherever it spills into. Yeah? That's the way that you can do it with traditional. Now, with AutoSpill, it's much different in terms of you don't actually need to do anything. Yeah, so you will assign your sample of interest on the parameter here on your empty channel that you find the highest autofluorescence, and in there, okay, that was it. You'll select AutoSpill, AutoSpread, and you don't need any to 0. So whatever there is positive will be subtracted from the subtracted from the rest. Okay. Okay, so some considerations for the autofluorescent extraction are that the unstained cell type, of course, should match the experimental cell type. Yeah, we don't want to have, use a different tissue to subtract the autofluorescence for another, right? We need to have either an homogeneous or an heterogeneous cell population within the sample. Yeah. It might, again, as I mentioned also earlier, if you have homogeneous, that would have better, if you have macrophages and you assign the same kind of sample for autofluorescent channel, you can then subtract the fluorescence for a specific population. If you have heterogeneous, it might differ from population to population. Okay, so we can look at this within FlowJo now. Okay, I think already we can see in FlowJo how it does that. Let me open a new workspace here. I have this workspace for AutoSpill. So basically what I have here is some samples from this, some samples from the paper. Yeah, so they're, if you want to find them, they're in the flowrepository.org. And basically here they wanted to showcase the AutoSpill algorithm and how it can remove autofluorescence. So they have only two samples here with a live dead staining, GFP cells. Okay, so there's only two parameters, and then the autofluorescence that we're interested. Yeah, those were highly autofluorescent cells. And if you want to check on that, you can, you can open your population, okay, right here. And what I would do as the first step is you could have a histogram here, okay, and you could see the different channels. Yeah, so if you, depending what you have, you could check the different channels where your population is mostly autofluorescent. Yeah? When you identify that, so this you should identify beforehand, of course, in the instrument, then you can use either AutoSpill or traditional compensation with your single thing controls. So I'll go here to my compensation group. Okay, so here I have some compensation controls, okay, pre-gated, and I'll show you how we can do the compensation. So before I do so, I'll just show you the results before the autofluorescence abstraction. Okay, so here what you're seeing is the GFP positive cells pre-gated on the live cells. And what we see from control versus the GFP mouse, okay, we have this autofluorescence. So you see this autofluorescence in the GFP. GFP is a quite common channel as well to have this autofluorescence. Yeah. And in the end, we had 9.3% of GFP positive cells based on this control. Okay. So, and now let's see how this would change when we subtract the autofluorescence. So I can go back here. I can open my compensation matrix, my compensation wizard. Okay. And now, okay, now in here, I have two channels only, which is the GFP and the live staining. But what I actually want is to add an empty channel. Yeah, so I can select here parameters, and then I can select choose parameters. And instead of having only the GFP and the live staining, I can also add one of the channels used for autofluorescence. Okay. In that case, we identified that this detector had the highest signal for the unstained sample. Okay, so I can select those three, just like that. Okay, so now I don't have any sample here, so I need to go right here and select my autofluorescent unstained sample. Okay, that's it. Right. From now on, I need to change my algorithm input to AutoSpill AutoSpread. Okay, because this is the way we're gonna do it in here. Okay. And we already pre-gated our sample. Okay, we have positive and negatives everywhere. And we also going to check the spectral button. Yeah, it makes, you see, it makes no difference to you. We have spectral. It will just take all the detectors. All you need to do is to press this spectral and you do the unmixing. It's nothing much, nothing more than that. Okay, and then I can select here view matrix and create a compensation matrix right here. Okay. 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 So now I created here my matrix. My matrix is ready, and I can apply it to all of my samples. I can just drag and drop this button, bring it in here, and then I can examine how my compensation looks like. Okay, so again, I remind you that this was without the subtraction. You see the autofluorescence here, and this is what we did now. Okay, so I try to subtract the autofluorescence. So you see now the GFP, yeah, the negatives, it goes here till 10 to the power of 4 approximately when we have the autofluorescence, yeah, and here we try to correct that, yeah, and here you see much clearer. Okay, if I put those side by side, you see much clearer and more well-defined population for the GFP, which is would be successful story. Okay. Let me check the chat box. Chat box is empty. Okay. Okay, so that's the compensation. Right. Let's go back to just to show you some metrics, because I mentioned earlier that we're going to go into some metrics. Okay. So why would we need metrics for compensation? Well, the first reason of needing metrics would be the spillover spread. So we mentioned earlier that after you compensate, you might see if two fluorochromes have very close spectral overlap, you might see this spread, this error. Yeah. And you can put numbers to it. Yeah, so the spillover spread was one of the first things that was described in such matrices, and you can calculate that in FlowJo. Yeah, So the spillover spread matrices are independent. That's very important because it's different than the total spread matrix. The spillover spread matrix is independent of the brightness of the controls, yeah? And it's mostly used to monitor the performance of the instrument that you have. Okay, so the value is mainly influenced by the spillover, the antigen density, and also the brightness. Okay, so in FlowJo 10.10, we can calculate easily this and export. Okay, so you can go after you have your matrix, either from traditional or from the AutoSpill algorithm. You can select the option SSM, you can visualize it as here, for example, in the compensation window, or you can export it in different formats. Yeah, and this can be calculated for both SSM, both traditional and AutoSpill. Okay. What we actually need here is, of course, low values. The low, the better. Okay. But again, if you have more than 10, you will notice more of spread. Okay. If you have less than 5, try to stay lower here to have better resolution. Yeah? And especially if you want to identify if in your panel the double positive are the the population you're interested, I would try to avoid having large values for markers that are co-expressed. Yeah, so you would need to be careful and go back to your panel design. Again, this traditional spillover spread matrix is independent of the brightness, so it's not really a good representation of the spillover that you will get in a set panel, but using having different dyes, for example, or different fluorochrome, or different antigen densities. It would change each time. Yeah? So even if you change that, if you can change your antigen density changes, your fluorochrome on different marker, etc., the number would be still the same. Yeah, so it doesn't account for those panel design, and it's not the best tool for panel design. Okay, the TSM on the other hand is not sorry. The TSM on the other hand, it doesn't account for this. It doesn't account for this normalization here, so it's doing it without normalization, so it's much better estimate of this spread. Yeah, so here the values are much higher in that case, and the TSM matrix is only calculated for the traditional algorithm. So there's no TSM matrix for AutoSpill, for the AutoSpill. Okay, so TSM is a good measure. It's a better measure than SSM for panel design. Yeah, because it's not normalizing the MFI. And if you decide to switch antibodies, if you say, for example, have CD3 on PE, and you make from CD3 to I don't know. CCR7 PE, okay, the total spread matrix would show you different results based on that, but the SSM would have exactly the same result. Yeah, so that's the main issue we bypass with the TSM. Other metrics that we have in FlowJo, and specifically on the latest version of FlowJo, is the CSM, which stands for Gaussian similarity matrix. So basically what this is basically comparing the signature of two fluorochromes, or two dyes. It's a value from 0 to 1, which means that if you have a value of 0, the two dyes are very different. Okay, so if you have, for example, a UV dye and a dye in the EPC, most likely those two would have a very low if we have high overlap or high spectral emission of those two dyes, you would have a value of 1. Yeah, so you would avoid having a very large value. Okay, so this you can inspect in FlowJo with different ways. So you can basically either use the spectral plots that you see here below, and you can put your different controls or different population and see how similar they are. And in there, you can also have this CSM chart. Yeah, so where you can have your controls in here and have those values exported. Yeah. Another way is you can find those values also in the chart from the compensation wizard. Okay, so it's also in there if you need it, or also in the BD spectrum viewer or in the BD research cloud. We have this function as well. Yeah, and this is more helpful for panel design than the rest. Yeah, and just the last metric here, I will touch ground too, and it comes mainly from a limitation that we have in the CSM matrix, yeah, on this similarity index. Yeah, and what was observed lately is that we might have, especially in large spectral panels, we might have some unmixing spreading error, which sometimes is not correctly identified with the similarity index. Take, for example, here, those two dyes. So we have high similarity pairs here in this panel, but actually, when we look at this into the, if we plot this against each other in a full panel or in a two-color panel, we see no significant spreading. On other occasions, you can see this spreading error only in the full panel, but not in the panel with two colors plus an empty channel. Yeah, so it's sometimes the similarity index can give either false positive results, okay, can a bit overestimate the error and give you a misleading results. So that's why some researchers within BD, they came up with a hotspot matrix. So I'll just show you an overview here, where basically the spectral hotspot matrix is a simple tool that predicts this unmixing error. It's a mixing dependent error for each fluorochrome in the context of a specific panel. So it's a much better and more targeted approach to find problematic regions in your panel. Yeah, so it will identify problematic regions. So here we have, for example, a large panel, and here are the problematic combination of fluorochromes that would cause a spread. Yeah, so there's a better correlation here of what you actually see as spread with what the hotspot matrix gives you as spread compared to the similarity index. It is available in the BD Research Cloud. So we have the following instruments here where you can measure more than 100 plus fluorochromes. It's for free to use, so you can create an essential account for free, and you can check your panel and see how you can optimize it. And it's important to mention that this is, you can check this even before you actually go for your panel design. Yeah, you don't need data here. If you want to calculate the SSM or the TSM matrix, you need first to do your experiment and then calculate. This you can do beforehand on your either on the research cloud or for your different samples, and then have an insight of if this panel would be a success or not. Yeah. So the website here would be bdresearchcloud.com. So you can feel free, you can give it a shot at your panel, at your fluorochromes, and look at what hotspot areas you should avoid in your future experiment. Okay. So just to the last step here, I'll go quickly. How to troubleshoot for compensation. It's a painful subject, I have to say. Seeing compensations like that. Yeah, so obviously you can see what is here a good compensation and what is bad compensation. But what is exactly the problem here? Yeah, this is something that we call overcompensation. Yeah, so we see that what we actually see is that we don't have the same MFI, okay, for the single positive CD8, okay, for CD19. Okay, so if you could put a value here for the median, you see that the negatives have a median approximately 10 to the power of 3, and here we're around 0. Yeah, so what we need for a correct compensation is to align that. Yeah. So the best practices published many years ago from Mario Roderer for a good compensation to avoid such occasion are the following. Yeah, we need our controls to be at least as bright or even brighter than the sun, but not less, yeah, than the compensation sample that will be applied to. The second is background fluorescence should be the same, and we mentioned that before. We cannot have beads with cells or different kinds of tissues within the same parameter. And finally, compensation controls must match the exact fluorochrome. And before doing an important experiment, if we use GFP mice for our experiment and we want to have a GFP in our compensation matrix, we need to have GFP cells, right? We don't want to make a single stain control using Alexa Fluor 488 or VT. We want to have exactly the same signature of fluorochrome, yeah? And that's very important. That's the source of the most errors we see there in data set where we apply compensation. Yeah? How we can check that in FlowJo? Easily. First of all, if the sample is bright or brighter, well, we can overlay them. Yeah, just use the layout editor, overlay the samples and see if you have the same brightness or less. Yeah, so here I have my sample, it's very bright. You see the positive here, but my single stain control is much less brighter. Yeah, so where do you set the gate? Right, that's a very tricky question there. So that's the first way to check that. The second, the background fluorescence, you can detect it easily on your compensation wizard. Yeah, so what you would see is this offset peaks for your positive and negative, where you see from your beads here, you have, of course, much less autofluorescence, and for your actual cells, your negative are higher. Yeah, so this mismatch must be corrected. Okay. And finally, you should use the same dye, lot and vendor. Some specific fluorochromes are more sensitive to from lot to lot, and you should always compare the observed spectral profile of controls to references profiles. So again, this is, you can look at your lab book and see, you have the same exact fluorochrome, which lot, and this is not something we can directly see in FlowJo, but this is something that needs to stay in your mind before creating your list of controls. Okay. So just to summarize, and just before I go to some questions here, compensation is really necessary. I hope I can convince you that it's necessary in multicolor flow cytometry. We cannot avoid it for now. And in order to calculate the abundance of each fluorochrome and correctly phenotype and place our gates. Methods do not include only the traditional, but now we have the spectral mixing, we have the gateless linear regression of AutoSpill and subtracting autofluorescence. We have also many different metrics I could show you. You will also receive an email very soon for the hotspot matrix with more details. And finally, controls. always about controls. We need appropriate controls and careful panel design so that we don't, go into situations where we have to edit the matrix or recalculate the complete matrix or rerun controls. Yeah, so the more careful we are when assigning those single stain controls in our experiment, the better would be the outcome. Okay. So let's see. I see a lot of questions in the chat box. So I'll try. I'll just try to go into here. Let me just. Okay. Where that? I'll go a bit up. Oh, that's a lot of questions. We do have some more time. Okay, I'll take it from Thomas' question. We have challenges generating reliable compensation for cells in double emulsion droplets. The droplets are approximately 60 millimeter in diameter with a colored cell inside. Our hypothesis is that our problems may come from the cells being out of focus when the droplet is hit by the laser. Could you comment on this? I need more info for this. I'm not sure I understand the question in there. I think it's something that needs more, more clarification. I don't know this exact technique you're using in there and how it has to do with the compensation. But I'll be happy, you can text me, you can send me an email, and I'm happy to provide more detail if we go through it in more detail. So another question, will applying a compensation matrix to a FlowJo workspace before acquisition give the same result as applying the same compensation matrix after acquiring the data? Before acquisition, what would you mean here? I mean, if we don't have the data, well, if you don't have the data yet, what you could do, I can show you right here. I guess you're referring to new samples that we want to apply a previously existing compensation matrix, but I'm not really sure. But remember that if you have a matrix, like I did here with the TFP, autofluorescence, and the light staining, you can save that matrix. Yeah, remember, you can save it as a matrix file on your desktop, for example. I'll save here my matrix on the desktop as an MTX file. Yeah. So now, if I rerun my samples or add new samples with the same panel, of course, same settings, everything, I can apply this matrix then on my samples. Now, after I apply this matrix, I still need to double check. It might be that there were some slight differences either in the sample preparation, antibody loads, anything. So it doesn't mean that your compensation will be exactly identical. That's why I would always suggest that you run your that you would run your compensation single stain controls every time it's fresh. Another question from Alexander, can we make the correct autofluorescent control from the GFP positive sample by exporting the negative population as a separate? I guess you wanted to raise a FCS file there. Well, That's a good idea. Yeah, so if you don't have any negative, exporting negative as a separate file, yeah, that should work. If those negative didn't pick up any dye, right, or if some GFP was somehow, if there were dead cells and and that happens also with staining. Yeah, if the cells tend to be sticky and capture some very little antibody and give some autofluorescence, but in that case, I think it should. I don't see a reason why it shouldn't work to set this as a negative. That's a very good idea. If you don't have, if you don't have the negative, that's a good way to bypass that. Yeah. You wrote there in CS5. Perfect. So the other question is, if I import already compensated data from Diva to FlowJo, does it change anything in the comp matrix value or population ratings when it's imported? No, yes, no. The exact identical matrix is transferred within FlowJo. And so another question is, if I understand correctly, negative population of single stain controls need to match the autofluorescence background. So it's better not to use beads. No, what I said is, if you use beads as the single universal negative, then you need to use beads for your positive as well. Yeah, it's just if you use bits, you cannot use cells. But if you use bits for negative, please use bits for positive as well. Yeah, so you reply to the answer to yes, if it is a quality control setting, a QC setting, where we can use the same compensation matrix for repetitive samples. Yes, that's exactly the way you could do that, to say the same matrix and apply it, but please be careful. You could do that, but depending on your panel, depending on how far away the different acquisitions were, you should pay some attention, to try to minimize the differences in this matrix. But in theory, it could be something that you can control. Okay. Okay. Instead of bits, can I use cells only? Absolutely. Only cells, unstained cells with stained cells. Or if it's the same tube and you stain, CD19 or something, okay. Some cells are CD19 negative, some are CD19 positive. So you have in the same tube the same, both populations. Yeah, so it's either only bits, positive and negative, or only cells, positive and negative. Okay. So I see no further questions here. So thank you all very much for staying. So a lot of you stayed. I'm very happy for that. I hope you learned something about compensation, and looking forward to any question. If you have, you can feel free to email me. Okay, my email address is, again, at the beginning, somewhere here. Okay, so here's my email address. If you have any additional questions, I'm very happy to take them. Thank you all very much for joining. Thank you.